The biological signal keeping you alive right now is hiding a catastrophic timeline error. Not a slight miscalculation, but a genetic sequence screaming from tens of thousands of years before human history even started. If your blood is Rh negative, you have probably heard the claim before from a documentary, a genealogy group, or someone speaking about mysterious origins and forgotten ancestors. The claim always sounds incredibly simple. Rh negative blood is unusually common among the Basques of northern Spain and southern France, a population that preserves some of Europe's oldest ancestry. Therefore, the logic dictates that Rh negative blood must come from Europe's first modern humans or even vanished species. At first glance, that sounds like a leap, but then the numbers start appearing, and suddenly the theory becomes much harder to ignore. In most populations around the world, Rh negative blood remains relatively uncommon, often falling far below 10%. Yet among the Basques, researchers documented frequencies reaching roughly 47.2%. Imagine walking into a room containing 100 randomly selected people from many parts of the world. Maybe 6 or 8 would be Rh negative. Walk into a room of 100 Basques and nearly 50 might carry it. The numbers alone make this a category-defining case for ancient DNA research. This is the classic story of an isolated group surviving an invisible environmental threat. It has happened before. It is happening here, within the human immune system. Outliers always make researchers curious, and the first temptation is obvious. Find the unusual trait, find the unusual people, connect the two, and call the case closed. Except history rarely works that way. The Basques are unusual for much more than their blood, and that is exactly where the mystery deepens. A second piece of the puzzle survived in plain sight through their language. If you travel across Europe, languages usually belong to recognizable families with visible connections, allowing linguists to trace relationships and reconstruct older branches. Then they reach Euskara, and the trail suddenly stops. Euskara stands alone with no accepted linguistic relatives, no surviving sister language, and no obvious family tree. The same population with extraordinarily high Rh negative frequencies also happened to speak Europe's only surviving pre-Indo-European language. One unusual trait might be coincidence, but two unusual traits demand serious attention. Then modern DNA studies arrived and provided fascinating, if imperfect, answers. The Neanderthal contradiction. I reckon the internet loves these stories because mystery sells. Millions of people have heard some version of the exact same narrative. Rh negative people are different, Neanderthals were different, humans and Neanderthals mixed, and therefore your rare blood type is an exotic import from the Ice Age. Human groups moved into Eurasia, Neanderthals already lived there, and that meeting happened roughly 50,000 to 60,000 years ago. We do not see a global evolutionary misunderstanding of this magnitude often. We may not see it again. Because the moment scientists finally decoded Neanderthal blood group genetics, the theory fell completely apart. The key ingredient was completely missing. Researchers examined blood group genes from multiple Neanderthal individuals, including ancient specimens from Denisova, Chigirskaya, and Vindhya. The expectation among believers was simple: find missing RhD genes and locate the ancestor of modern Rh negative blood. Instead, scientists found Neanderthals were Rh positive homozygotes. Rh negative blood depends largely on the absence of a working RhD gene, often through a deletion. Neanderthals actually had 2 copies of the normal gene, meaning there was absolutely no missing version hiding in the background waiting to emerge later. You cannot inherit a missing genetic instruction from ancestors who carried the gene normally. If a grandparent never owned a blue car, they cannot pass that blue car down to you. The theory does not merely become weaker, it completely loses its foundation. To be fair, the genetic evidence gets even stranger. A 2021 study examining Neanderthal and Denisovan blood groups identified a rare, unique Rh allele that is largely absent from modern humans. Only one Aboriginal Australian and one Papuan have been reported carrying something even remotely similar. Instead of becoming common Rh negative blood, the Neanderthal variant mostly disappeared entirely. The evolutionary path runs actively away from modern Rh negative blood, not toward it. Furthermore, older claims described Neanderthals as mostly blood type O, but ancient genomes revealed they actually displayed A, B, and O blood groups. One Siberian Neanderthal carried type A, while a Croatian Neanderthal carried type B. If you are looking for the biological equivalent of a geographic impossibility, this is closer than it should be. The casual version is the one most social media documentaries ran. The version that matters is the one the geneticists actually sequence from ancient bone fragments. The timeline collapse. And this is where the evidence gets really interesting. The first major crack in the popular narrative actually appears in Africa. Everything in the folklore points north toward Ice Age landscapes. But genetics quietly points millions of years earlier to Africa. The common RHD deletion associated with Rh negative blood appears as a prevalent D negative haplotype in African populations. That single brutal observation creates a catastrophic problem because Neanderthals never lived in Africa. If the same deletion already existed before migrations into Europe, Neanderthals absolutely cannot be its source. If the Neanderthal theory is true, your blood type is an exotic import. If the data is true, your entire ancestral survival hinges on an ancient African mutation. There is no middle ground. The timeline is not slightly wrong, it is catastrophically wrong. Imagine trying to explain a Roman coin by confidently claiming it came from World War II. The dates simply refuse to fit together. Evidence indicates the mutation arose long before modern humans settled Europe, long before agriculture, and long before any meeting between modern humans and Neanderthals. Most who hear about rare blood types miss the African origin entirely. You and I do not. Researchers also noticed that Neanderthal Rh variants remained remarkably stable for around 80,000 years, while human Rh variants changed much more rapidly after populations left Africa. The spotlight fundamentally shifts away from inheritance from another species and lands squarely on evolution within our own. The geographic mismatch. Look at the population numbers across the globe. They are absolutely brutal. If Neanderthal ancestry caused Rh negative blood, then people with more Neanderthal DNA should naturally show more Rh negative blood. East Asian populations in China and Japan carry Neanderthal ancestry that is broadly comparable to Europeans. Yet their Rh negative rates hover extremely low, often around 1 to 2%. Basques can show around 1 in 3 Rh negative, while Chinese and Japanese populations show around 1 in 50. If Neanderthal ancestry were the driver, those numbers should not split like that. They should move together. Not all Rh negative results around the world even come from the exact same genetic cause. In some East Asian populations, RHD negative individuals carry forms like DEL variants. A DEL variant can make the D antigen extremely weak or incredibly hard to detect, meaning the simple label of Rh negative actually hides entirely different genetic pathways. Different populations can reach Rh negative status through wildly different historical mechanisms. Imagine finding 3 houses painted red and assuming the paint came from the exact same factory, only to discover one used oil, another used acrylic, and the third used a completely different pigment. The color perfectly matched, but the origin definitively did not. Certain Berber communities in North Africa and some Jewish populations also display elevated frequencies of specific Rh negative variants. When 3 dots appear close together, the brain desperately wants to draw a line connecting them to ancient Hebrews, Sumerians, or lost homelands. But shared percentages do not automatically equal shared ancestry. Nature constantly repeats successful biological solutions. Imagine 2 distant islands that both develop houses on stilts because local floods produce the exact same architectural solution independently. If 2 populations live under similar environmental pressures, similar genetic traits can easily become more common without sharing a recent ancestor. The evolutionary paradox. This brings us to the actual danger pulsing through the bloodline. Evolution is usually ruthless, slowly removing traits that create a severe reproductive disadvantage. Before modern medicine, an Rh negative mother carrying an Rh positive baby could develop aggressive antibodies. Think of antibodies as tiny biological security guards trained to recognize a foreign presence and violently attack. During later pregnancies, the mother's immune system could target the baby's red blood cells, resulting in severe illness or even death. The blood type that can routinely kill a baby should have completely disappeared from the human gene pool long ago. Instead, it stubbornly shows up in roughly 15% of Europeans and around 6% of humanity globally. Why did it survive? Real life is remarkably messier than perfect biological engineering. Evolution simply cares about survival, and sometimes survival involves accepting one horrible problem because another problem is substantially worse. Imagine carrying a heavy, annoying umbrella every single day. It feels incredibly inconvenient until the alternative is getting violently soaked in a storm every afternoon. Genes operate the exact same way. If the underlying benefits are greater, a costly trait remains locked in the population. Some researchers explored whether Rh negative frequencies might relate to crucial protection against environmental challenges like Toxoplasma gondii. This parasite is surprisingly common, still infecting huge numbers of people worldwide and causing serious effects in certain situations. Research suggests Rh negative status may fundamentally influence how people respond to toxoplasmosis, affecting its consequences rather than completely preventing infection. Think about isolated ancient communities surviving with no antibiotics, no modern hospitals, and absolutely no prenatal care. A tiny survival advantage against an invisible parasite could strongly ripple across thousands of generations. One child survives, then has children, then grandchildren, accumulating tiny differences until a gene that should have disappeared is fiercely present thousands of years later. The sheer scale of the genetic deletion spreading from Africa across the globe defies simple comprehension. The power of isolation. This is not about an internet myth. It is about a devastating reproductive cost that shaped who lived and who died. Everyone involved knows the difference. Geography drastically changes the odds of survival. The Basques provide one of the most undeniable examples of genetic drift. Located in mountainous regions spanning northern Spain and southwestern France, they maintained genetic continuity stretching back thousands of years. Mountain valleys act like biological filters, slowing human movement and making genetic preservation highly likely. Imagine a village completely hidden in a valley where marriage occurs mostly within the immediate community and new arrivals are historically uncommon. Genetic isolation acts exactly like a magnifying glass, turning rare traits into dominant features simply because the population remains relatively closed. The Irish also show elevated rates compared with the global average because island populations heavily limit mixing. A recessive trait needs 2 copies of the variant to be visibly Rh negative, which normally makes the trait incredibly hard to maintain. But within the strict boundaries of isolation, a recessive gene can persist longer, and eventually become a deeply recognizable feature. Of all the genetic mysteries of the genomic era, the Rh-negative origin is the one that completely broke our timeline. Modern DNA research dramatically complicated the image of the Basques as living fossils. One major study discovered severe evidence that Basque male lineages actually experienced extensive large-scale replacement during Bronze Age migrations between roughly 2200 and 1800 BCE. Yet, simultaneously, maternal lineages showed far deeper continuity, extending back to ancient European roots. Human populations change constantly as genes mix, but resilient traces of older ancestry securely survive through all the chaos. Remove the Basque population bottleneck from the Ice Age story, and the European genetic story simply does not survive. The blood archive. Every generation has its enduring biological mystery. The Rh system is the modern version of a map written in blood, showing farmers moving across Europe, hunter-gatherers adapting, and steppe groups crossing enormous distances. Early European farmers appear to have carried relatively low Rh-negative frequencies. Hunter-gatherers appeared moderately higher, while steppe populations registered higher still. Modern Europeans heavily descend from all three groups in wildly varying proportions. Their descendants relentlessly mixed, separated, and then mixed again over centuries. Through all that chaotic movement, Rh-negative variants stubbornly survived. Then, human intervention violently rewrote the biological rules. In 1968, RhoGAM finally became available. A simple medical injection could suddenly prevent the mother's immune system from becoming fatally sensitized. The severe reproductive danger that threatened thousands of future pregnancies could finally be avoided. For thousands of years, blind biology wrote the rules, and then modern medicine permanently edited them. A deeply reproductive disadvantage that persisted for brutal centuries immediately became far less historically important. That rapid medical intervention explains exactly why the trait remains strongly common today, effectively splitting the history of the blood type into ancient evolutionary gambles and modern clinical victories. There is no one else who could have endured this. Your blood type is a brilliant genetic archive recording a long chain of intense human events. It represents a rare mutation, an aggressive population split, an isolated valley, an exhausting migration, and an infant who miraculously survived when countless others absolutely did not. Evolution is not finished. The next genomic survey decides whether these DEL variants reveal even deeper lineages or something completely unexpected. What the scientific community does next with ancient blood group analysis is what people will remember. The final determination has not been made yet, and here we are. The biological story does not end with RhoGAM in 1968. It does not end soon. We are in the middle of it. If one small genetic marker can accurately preserve desperate traces of massive population movements thousands of years old, what other stories are sitting quietly inside your DNA right now, waiting for someone to finally recognize them? We will be here when they finally surface.